Today we're talking about enzymes. Enzymes are a very special group of proteins. The majority of enzymes are. So about 99% of enzymes are made of proteins. And so if we go all the way back, that means that since proteins are made of amino acids, enzymes are actually made of amino acids also. The reason that the majority of enzymes are made of proteins goes back to how those amino acids interact and then fold into shapes. And that's going to be a really important characteristic of enzymes, is that they are a very, very specific shape. Enzymes are often named with the ending "ase." The majority of them are. So if you see something that ends in "ase," it's usually an enzyme. So we might see catalase. We might see DNA polymerase. All of these, just recognizing the "ase" is going to tell us that it's an enzyme. And this is kind of what you saw before when you look at how an enzyme works. Really what an enzyme is doing is helping with some sort of reaction. And so the shape is very important because it helps with specific reactions. So at this point we want to know that enzymes are usually made of proteins because that's going to relate to the shape. We can recognize an enzyme by "ase or by the fact that it performs a specific function. That's what we'll talk about next. What is an enzyme? What does it do? Well, they have four main characteristics. The first is that they are considered biological catalysts. So let's break this down. Biological meaning they are found in living organisms. So there are lots of different catalysts. Catalysts speed up reactions. That's what a catalyst does. Catalyzes a reaction. Helps it happen faster. And we'll talk about how in just a second, this idea of activation energy. But enzymes are considered biological catalysts because they're found in living organisms. And a lot of this is because it's a protein. And this protein is made by the cell. And so one thing that we'll see is that enzymes are found in unity, meaning some living organisms have all the same enzymes, and they also produce diversity. Even within a single organism, we might only find an enzyme being produced by one type of cell. So understanding that they're found in living organisms, different living organisms can have similar enzymes or different enzymes. And that gives us a little bit of idea about how related organisms are. So let's talk a little bit more about this idea of catalyst. How does it speed up a reaction? Well, when we go back and look at what a reaction is in chemistry, there's this idea of energy. Usually when we talk about a reaction, we are talking about building or breaking bonds. Well, what is a bond? Remember, a bond is between electrons. And there's energy between those electrons. So if I'm building or breaking bonds, I am involving energy. Some of these reactions will release energy. Some of these reactions will require energy. So how do we get the reaction started, right? We've got this reactant and it's stable. And how do we change it? And in order to get it started, we call that the activation energy. So this might be like, consider starting a fire. Once you get a fire started, it's releasing energy. But you have to provide some sort of friction or force to get that fire started. And enzymes are going to help reduce the amount of energy needed. And And a lot of times what enzymes are actually doing is they are aligning the substrates to kind of encourage a reaction. What the enzyme is doing is really providing a location for this reaction to happen so that it maximizes the probability of the reaction. So having two things separate and bringing them together, that's kind of what the enzyme does, is encourages that reaction to happen. So we want to be able to understand that without an enzyme, we've got uncatalyzed and catalyzed. And we can see that uncatalyzed, I have to put a lot more energy in. I end up at the same spot. I started at the same spot. But without an enzyme, I need a lot more energy to be put in. And we call that the activation energy. So we will say that enzymes lower the activation energy. They allow the reaction to happen faster. Some of these reactions are going to be breaking down. Some of these reactions are going to be building. For the majority of reactions, if I release energy, this is what's known as an exergonic reaction, and that's releasing energy, a lot of times as heat. And these are usually breakdowns or catabolism, digestion, reactions where energy usually needs to be put in, where energy is being supplied or consumed. These are usually, this picture isn't showing it, but these are usually synthesis reactions. These are called endergonic, E-N-D-E-R, ender, putting energy in, endergonic reactions. And these are usually going to be building reactions. So that's an important part because enzymes can, in theory, either build or break down. But the next thing we're going to see is that enzymes are actually specific. They can't go forwards and backwards. They really only do one thing. And this has to do with the fact that enzymes have a specific shape. That specific shape comes from the folding of the protein. And so we can see here, it kind of looks like a blob, but what we actually see is something called the active site. And the active site is the location where the substrate enters. And this is the part of the enzyme that the shape is primarily, number one, primarily important. If this shape doesn't fit the substrate, the reaction will not occur. So substrate is actually the word, remember, that we use to describe what we start with. The substrate is what we start with. The substrate has to fit into that active site. And it's sometimes called a lock and key, and that's just to describe that it is specific. But more advanced versions have actually shown that we usually have what's called induced fit, meaning that once the substrate fits, and then there is a conformational change. And conformation means shape change. And this is where the enzyme folds closer to the substrate. And so when we talk about a substrate fitting, we need to think of the properties of the enzyme and the properties of the substrate. So when we think about this substrate, it needs to be attracted to the enzyme site, to the active site, So it needs to have similar properties. So we need to talk about polarity. we're not just talking about physical shape of the molecule, we'd also talk about polarity, hydrophobic, hydrophilic, and then that enzyme will fold around it, and we call that induced fit. And now we have what's known as the enzyme substrate complex. After the reaction is over, the enzyme will actually open up and release the products, and now I'm back to the original shape of the active site. So the first property we looked at is that they're catalysts. They speed up reactions by bringing the substrate in and helping it change. The second characteristic is this idea of specific shape. We're going to talk a little bit later about other parts of the enzyme that might need to be a specific shape as well, called allosteric sites. But right now we're focusing on this idea of the active site, where the action happens. So to take a break, we want to review about why is it that most enzymes would be proteins? What is the relationship between the enzyme, the substrate, and the product? Describe the difference between an anabolic reaction and a catabolic reaction. Catabolic is going to be the one that we talk about in terms of breaking down. So anabolic would be the opposite. It would be a synthesis reaction. So taking a second to kind of process this part of enzymes, and then the next part that we'll talk about a little later in the next section will be how enzymes are reusable and how enzymes can actually be damaged. But we're going to stop here for now.